Hello and welcome everyone once again to the biology class of Fun Scholar. So today we are going to discuss a new chapter, that is the cell. This chapter that we are going to discuss is there in the 6th standard of ICSE as well as in the 8th standard of CBSE. So let's begin the discussion. So here you can see cell is known as we will study the structure and the functions of cell. But before going into that, I would like to let all of you know that cell is the preliminary idea that we all need to have whenever we are going to study about biology, because our study about biology will be incomplete if we don't know about the basic foundation, and that is laid by a single cell only. So therefore cell, without actually knowing about the cell, we cannot know about all the other functions associated with every organ of our body or every part of our biological system, because cell is an integral part and the building block of all these biological system as a whole. So therefore cell is very much important for discussion. That is why it is always given the first priority chapter wise and also knowledge wise. So whenever we have to understand the cell, we can compare it by the brick of a building. For example, how long or whatever be the height of the building, or whatever be the actually the large or the magnitude of a building, but that cannot be built until and unless the bricks which are laid, that are actually laid in a proper manner. So therefore each brick, if we compare that, is like the cell of the whole building that actually lays the foundation of the whole structure. Similarly, cell lays the foundation of the structure of the living organisms in the living world. So cell is very important to be studied. And in the structure of the cell, we'll get to know about some of the structural components without which the cell cannot exist altogether, and also we'll learn about certain cell organelles also which assist the cell in many other functions. Now these cell organelles, they are actually helping in the manner of division of labor, that we'll understand subsequently in this chapter. So now is the video time, and we'll be first watching a video on cell.
Have you ever played Lego? What have you built with it? Houses, animals. Many Lego pieces, when put together, can form amazing structures. You can make small structures or even huge ones. But no matter what the size of the structure is, its basic unit remains the same. A single Lego piece or a Lego brick. A single Lego brick is the basic unit of these structures. Lego pieces, when interlocked with each other, create different structures of different shapes. Do you think living organisms are also made up of something? Yes, they are. Living organisms are made up of cells. Cells are the basic units of an organism and are called the building blocks of life. Cells, like Lego pieces, give a structure to the body of an organism. Several cells together form a human hand, leg, face, and other parts of the body. Therefore, they are the structural units of the body. However, unlike Lego pieces, cells of living organisms carry out all the basic functions of the body like digestion, respiration, etc. That means they are also the functional units of the body. Cells not only give the body its structure, but also also carry out the functions of the body. Let's talk about the structure first. The cells give a structure to the body. What do we mean by a structure? Do all animals look alike? The tiger looks different from a butterfly. It looks different from a fish. What makes them look so different? It is the cells that contribute towards the structure. The cells build up the body of different organisms in different ways. The cells also carry out different functions like flying in birds, swimming in fish, and so on. So what we learned so far is that the cell is the basic structural and functional unit of the body. Now take a look at yourself in the mirror. Can you see the cells in your body? No? Move a step closer then. Do you see your cells now? No. How about another step closer? You still cannot see the cells in your body? Why is this so? Human cells are minuscule and they cannot be seen by the naked eye. Now look at this picture. It is the egg of a hen. Can you see the yolk of the egg? The yellow part of the egg? Do you know that this is one single cell? Yes, the yolk is one single cell. Wait, we can see this cell. On the other hand, we cannot see human body cells. Yes, some cells cannot be seen by the unaided eye, while others are large enough to be seen. Do you know there are some living organisms which are made up of just one cell? This single cell carries out all the functions of the body. For example, the amoeba is a single-celled organism. This single cell carries out all the functions like ingestion of food, digestion, excretion, and even movement. What is such an organism called? Come on, you can guess. We call it a unicellular organism. On the other hand, human beings are multicellular organisms. We are not made up of a single cell. Our body has different types of cells to carry out different functions. But do you know multicellular organisms have emerged from a single cell? Single cells multiply and divide to form tissues. Each tissue is made up of similar cells which form a group. These tissues then join together to form organs, and several organs together make up an organ system. A multicellular organism is made up of several such organ systems. Human body has the digestive system, the respiratory system, the excretory system, and many more. All these systems are made up of cells. Therefore, the cell is the fundamental unit of life. Isn't that amazing? Now, what about the size and shape of the cells? Are all the cells of the same size and shape? Here are a few images of cells. This tiny one is a bacterial cell. Can you see this elongated cell? It has got a few wires like projection as well at both ends. It is called a nerve cell. And this one here is the yolk of an ostrich's egg. Shall we measure these? The size of the bacterial cell is 0.5 micrometers and it is spherical in shape. The nerve cell is long and the width must be somewhere around 100 microns or 0.1 millimeter. It is a long elongated cell. What about the yolk? It is somewhat spherical and measures 170 millimeters in length. The width is 130 millimeters. So it's clear that the shapes and sizes of the cells are not always the same. The bacterial cell is the smallest living cell so far. On the other hand, the human nerve cell is the longest cell in the human body. What about the ostrich's yolk? It is the largest single cell found so far. Cells are of different shapes and sizes in different organisms. But does that mean the size of an animal and the cell size are interrelated? Let us explore this idea. Look at these two animals, an elephant and a fish. Do you think the elephant will have huge cells because its body is huge? Or does the fish have very tiny little cells because it has a small body? No, that's not the case. The size of the cells do not correspond to the size of the body of an organism. The shape and size of a cell is determined by its function. For example, the nerve cell is elongated because it has to carry impulses from different parts of the body to the brain. So it's like joining two ends to carry messages, just like a telephone wire. Each cell has a specific function. However, most cells are microscopic in nature. Then how do we know that cells exist? What can help us spot them? The microscope. The microscope magnifies the specimens on the slide and helps us to study even the minuscule elements of nature. We will study this in detail in the videos that follow.
So from this video, we came to know about many things, right? What are they? Let's summarize one by one. First of all, we have come to know that cell is the basic structural and functional unit. Why? Because I have already told in the beginning that without the cell, no structure, however small or big it is, cannot be built. I am talking about the structure of any living organism which actually are present on the surface or on the face of the earth. So therefore, it is the basic structural unit which helps us to build all the living structures, and functional unit because the cell is independent enough to carry out all the functions so that it can survive in this living world. So it is not dependent on any other cell. That is why functionally and structurally it is unique, it is independent, and also it lays the foundation for organization. That means the hierarchy of organization that you have also watched in the video, that after cell the hierarchy that actually is followed till an organism is built, the cell helps us to build them bit by bit. Therefore, it is the basic structural and functional unit. Now, you can ask me also, cell has also got cell organelles. That we'll study in detail later on in this chapter only. But then, why do you say that cell is the basic structural unit? Because cell can be further divided also. It is because the cell definitely can be further divided, but those organelles cannot exist independently like a single cell. A single cell can exist in the environment independently because if it has got all the cell organelles working or performing correctly, then it will be able to survive in the environment without any problem. But if we just leave any cell organelle in the environment, then that organelle on its own cannot survive in the external environment. It can only function when it is present inside the cell or in the cytoplasm. So that is why the cell along with the cell organelles as a whole is the first fundamental unit that can exist independently and that can perform all the functions of living organisms as a whole. So therefore, it is the structural as well as the functional unit of life. Now, the second point was that this was the first point, that structural and functional unit. Second point was about the hierarchy of living organisms. So, how hierarchy of living organisms that is arranged? So, it is arranged by it. The starting point is obviously the cell. So, the cell. Then the cells of similar type and of similar function will aggregate together to form tissue. Then tissues of same type will aggregate to form organ. Then the organs which actually they help in a certain particular function of the body, that will aggregate to form the organ system. And finally, all the organ systems together will give rise to a whole organism. Right. So this is the idea of hierarchy in living organisms. Okay. So now tell me, if the foundation step, that is the cell, it is missing, then is this hierarchy or the arrangement possible? I guess the answer is very obvious. It is not. So that is why the cell also lays the foundation for hierarchy in the living organism. And the third point that we have learned from the video is of the variety in cell shape and size. Right. So cells come in different shapes and sizes based on their function. So according to their function, we can get cells of different shapes and different sizes. That already have been discussed. Like neuron, it is long thread-like because it has to conduct the neural impulses. Then we have got the RBCs, which are biconcave disc shaped because it contains the hemoglobin also inside it, which is also known as the oxygen carrier. So then we have got the WBCs, which does not have a definite shape, which is known as the amoeboid shape, because it has to engulf the pathogens and the other impurities entering our body from the environment. So according to the different functions, cells can be of different shapes and sizes. So these are the 3 points that have been primarily discussed in the video. So now let's move on to the next part. So living organisms are made up of different types of cells of different shapes and sizes that we have just now seen. Now coming to the point, who discovered the cell? Cell had been there always, but somebody was there to make the world aware that yes, there is something called cell that exists and on which the whole living world is actually dependent. So therefore, let's know about that person or people who actually gave the idea of cell to the whole world. Now let's watch this video.
In the last video, we studied how tiny the cells are. If they are so tiny, how did we find out they existed? Let us go back in time. In the year 1665, we had a person named Robert Hooke. He created a self-designed instrument which in today's terminology we call a microscope. This microscope is a very crude version of the present-day microscope. Let us see how it worked. The microscope used a flame in order to provide light to see the specimen. What do you think Robert Hooke observed under this microscope? Can you see this tree? This is called its bark. He had a thinner outer layer of the bark of the tree examined under this microscope. To his amazement, he saw a cluster of spaces which looked like this. What did he think when he looked at these spaces? For Hooke, these resembled the tiny rooms where the monks lived. So he called them cells. A cell means a tiny room. That's how we got the name. What else do these resemble? Yes, they also look like a honeycomb. Can you see each cell is separated from each other? There are these walls that separate each cell from the other cell. However, this microscope could not give detailed images of each cell and its structure. The cell space within the walls was empty. Do you know why? It's because these were dead cells. Then who was the first one to view a live cell with a better microscope? Along came Anton von Leeuwenhoek in the year 1671. He created the first simple microscope. You would probably think he was a scientist to have created a microscope, right? No, he wasn't. He was a textile merchant and he wished to study the quality of his threads. Thus, he used lenses to create a microscope which looked like this. The observer had to see through this lens, and the specimen was kept on this pointed piece, and this observation had to be done by facing the sun. Anton van Leeuwenhoek, by the way, is known as the father of microbiology. He was the first one to study the protozoa. He gave the individual cells a fancy name. Do you know what he called them? He called them animalcules. As time passed by, it is believed that Leeuwenhoek was able to create microscopes with a magnification of about 2 to 500 times the original one. So Anton Leeuwenhoek was the first to study the live cells. Until then, the details of the structure of these minute organisms weren't available. Once the discovery of the live cell was made, people wanted to know more about its structure. Robert Brown was the first one who discovered the nucleus. The modern microscopes have created new possibilities for the study of the structure of the cell. With newer information day after day, scientists started studying different cells. In the 1830s, we had Matthias Schleiden, a German botanist who studied plant cells. And Theodor Schwann, in the same decade, was studying the animal cells. Both of them realized in their comparative study that plants and animals are made up of cells and cell products. But how did these cells originate? Where did they come from? This was answered by Rudolf Virchow in the year 1885. According to him, cells divide and form new cells. Therefore, new cells are formed from pre-existing cells. He then modified the hypothesis of Schleiden and Schwann, and together they proposed the cell theory. The cell theory makes two important statements. The first is that all living organisms are composed of cells and products of cells. Secondly, all cells arise from pre-existing cells. Yes, all living things are composed of cells and its products. And all cells are formed from pre-existing cells. With the invention of the electron microscope, one could now see the inner structure of the cell. Have you ever observed the workers at a construction site? Every person has been assigned different functions. Our body, too, has assigned different functions for different organs and organ systems. This is called as the division of labor. In a similar manner, the tiny cells, too, have organelles within it. These organelles have specific functions. We will study this in our next video.
Okay, so now from this video as well, we have got to know some of the very relevant points regarding the discovery or the history of cell studies. So therefore, what are the points? Let's summarize again. First of all, we came to know about Robert Hooke and Anton von Leeuwenhoek. So Robert Hooke was the he was the first scientist. He was British. So therefore, he was the first scientist who actually observed the dead cells in the year 1665. Right. First of all, okay. Secondly, he observed the in 1671 observed live cells. What type of live cells? Protozoa. It is a single celled organism and he called them animalcules. Right, he coined the term animalcules, and he actually observed these cells by inventing simple microscope. By inventing simple microscope, right? So Robert Hooke first observed the dead cells in the year 1665, and then live cells were observed by Anton von Leeuwenhoek, and he was not the scientist, he was the Dutch merchant of textile. He used the simple microscope to study the fiber quality of the textile which was produced in the factory. But he is the person who is known as the father of microbiology. Why? Father of microbiology. So I can write AVL in short for now. That is Anton von Leeuwenhoek, because he was the person who first observed the live cells with the help of simple microscope. Later on, that microscope underwent many changes, and today we have got the modern compound microscope. But at that time, when nothing was invented, so he was the person who took the initiative to observe cells. That is why he is known as the father of microbiology. So, and one more thing we came to know, that is the cell theory. What is cell theory? That means cell theory basically states about the origin of the cell and the nature of the cells. So you have already watched in the video that the cell theory postulates, which are mainly that all the cells arise from the pre-existing cells, and all the living animals are made up of, or the living organisms are made up of cells. So the living organisms were broadly categorized at that time, back at the year. This cell theory first came in 1839. This was the time when Scladen, Matthias Scladen, and Theodore Schwann, they were the two scientists who developed the cell theory first. And that was the time when Scladen studied the plant cells, and Schwann studied the animal cells. So, but they were contemporary to each other, and they found the same observation, similar observation, that both in the plant cell as well as in the animal cells, the organisms were made up of that tiny cells, very microscopic, very small, tiny cells. So they came to the conclusion that all living organisms are made up of cells, and that is why cell is the basic structural and functional unit of life. These were the two cell theories proposed by both of them. Now, what happened afterwards? Rudolf Virchow, in the year 1855, so he added one more statement or postulate to this cell theory, that all the cells arise from the pre-existing cells by cell division. Now cell division also is of two types that we will study later on, but as of now we need to understand that cells, actually every new cell arises from the previous cell only by the process of cell division. So therefore, these are the three basic postulates of cell theory proposed by Scladen, Schwann, and Rudolf Virchow. See, this is all that we have discussed. Now, one more thing. When Robert Hooke observed the dead cells, so they in the cork cells of the bark of a tree. So the cork cells are dead cells. They have lost the protoplasm and they are no more alive. And that is the reason, that is why he observed that the cells were hollow. They had a definite shape, like rough hexagonal shape, but they had nothing within them. That is, they were hollow because they were dead cells. Now we know that most of the tissues of the plant cells are dead because they are stationary and do not require so much energy for their regular activities like animals. So therefore most of their cells are dead, and also that helps them, that gives an advantage to the plants for less maintenance. Also they do not have to acquire more nutrients for the maintenance of the dead cells. So that is an advantage. But at that time it was not discovered why the cells were dead and why the structure were missing inside the cells, and why the only the outer skin, it looked just like the outer skin of the cells there that are attached together. So he observed the dead cells, and later on in the year 1671, Anton von Leeuwenhoek was the person, with the help of microscope, he then observed, as I told you, the live cell. That is why he is known as the father of microbiology. Now, we have been discussing about dead cell, live cell. Now, we need to know what is the difference between dead cell and live cell. What are the features which are there in a live cell and missing in a dead cell? So, we need to know that a cell is considered living when it contains protoplasm. What is this protoplasm? Protoplasm is the jelly-like substance inside the cell. So, jelly-like substance means it is the matrix, we can say. It is the matrix of the cell. It is the sum total of cytoplasm and the nucleus. Because within the nucleus, we have the matrix of the nucleus itself, which is known as the nucleoplasm. So, we can say the sum total of cytoplasm and nucleoplasm, that gives rise to protoplasm, and the cells which lack protoplasm are dead cells, but the cells which contain them are the living cells. Now, cytology is the branch of biology that deals with the study of cells. Cyto means cell and logy means study, right? So when we club the two words together, we get cytology, that means the study of the cells. And the study of the cells has been facilitated by the invention of the modern versions or the most advanced versions of the microscope. So nowadays we have the electron microscope, so therefore which helps us to study the detailed structure which are at the very I can say that they are so small that we cannot even imagine that they exist in our living world. So therefore cell itself is not visible to the naked eyes, or we all know that most of the cells are microscopic, we have already heard from the video, but still the parts of the cells, and also nowadays we can also understand the parts of the cell organelles also. So that is a great achievement and a milestone that has been achieved by the biologists so far. So microscope and cell study, we will study about the microscope now a bit. In case of simple microscope, I already told, which was used by Anton von Leeuwenhoek to study the live cells. What was the components? It was composed of a single biconvex lens. That means biconvex lens looks like this, that biconvex on both the sides. So therefore a single biconvex lens which used which was used to study or magnifying the image of the cell. But nowadays what happens that compound microscope is basically used in the laboratories especially, and also in all the educational institution we use the compound microscope. Now this microscope is known as the compound microscope because of this point, uses multiple lenses for better magnification, like multiple lenses like the eyepiece which remains close to the eye, with which we observe the specimen. That is why it is known as eyepiece. And all the lenses are biconvex only. So this is the eyepiece. Then we have got the objective lens which is present near to the object on the stage. So that is why the name objective lens. Now objective lens are either 3 or 4. So it depends that with which power you would like to observe the specimen. So we start with the low power of the objective first, then we to focus, and then we move on to the higher powers. This is the basic thumb rule for setting the microscope in order to focus the specimen. So therefore, now the objective lenses we have got the 10x means the object will be magnified 10 times, then 45x that means the object will be magnified 45 times, and so on, and also 100x that means the object will be multiplied or magnified 100 times. Now how do you calculate the magnification power of a compound microscope? It is the product of the magnification of the eyepiece and the objective that we are using to observe the specimen. The eyepiece has the fixed, we can say a typical type of power of observation or power of magnification that is usually set at 10x, and if we multiply that with that of the objective lens power, either 10x then we will get 100 times magnification, 10 into 10, or it is 10 into 45, then we will get 450 times magnification, or it is 100, then we will get 1000 times magnification, right? So this is how we calculate the magnification of a compound microscope, and since we use here majorly two convex lenses, so the magnification that we get is actually the sum total, or we get as the power of the magnification of both the lenses. So that is why it is known as compound microscope. Because now coming to the cells which vary in their number, shapes and sizes. So in the number, cells can vary from a single cell which is also known to be unicellular to multicellular. Right? We are multicellular, plants, animals, all are multicellular. So unicellular organisms are also there. You can see here amoeba, chlamydomonas, then we have paramecium. So these are made up of a single cell, and that single cell is capable enough to carry out all the life processes of that particular organism, right? So therefore this is their body and is made up of one cell only. So that is why they are known as unicellular organism. Now cell shapes and their functions. As I told you that the shapes that differ or vary depending upon the functions that the cells perform. So spherical cells, I already told you, the red blood cells, they are biconcave disc shaped. Biconcave disc shaped. You can see the middle portion is narrow, right? It is narrow like a pit, and then the periphery region is swollen. So that is the biconcave on both sides, and then so it actually contains the hemoglobin, and it helps in the transportation of oxygen. Then oval cell shape is that of chlamydomonas. It is a unicellular alga. Then we have cylindrical shape, that is the Spirogyra. It is multicellular. It is also a type of algae and it is multicellular. And green means it obviously will contain chlorophyll and it can conduct the process of photosynthesis. So therefore Chlamydomonas and Spirogyra both can be counted under the category of autotrophs. So then we have got amoeboid cells, that means with no particular shape, it can change its shape as and when required. Just like amoeba who can change the shape and form pseudopodia for engulfing the food particles and also for discharging the waste products after digestion is complete. Similarly, WBCs I already told, they also can change their shapes and engulf the pathogens when they enter our body. Then we have the cubical or rectangular shape, that means the plant cells which are sort of hexagonal or rectangular, we can say, right? Then slipper shaped, just like the paramecium. This is the paramecium. It looks like the slipper which we wear in our feet. So therefore, that is known as the slipper shaped animalcule because this name was given by Anton von Leeuwenhoek, who discovered it with the help of his simple microscope. And we also have got spindle shaped, that means the ends are thin or tapered and they are in the middle it is broad. See, this shape is known as the spindle shaped. Also we have the wire like projections such as the neuron that you have already watched. This sperm cell is also having a tail for swimming and moving to the female reproductive tract. So every cell shape has a reason behind it. Now coming to the examples of different cell shapes for the different reasons again. So human sperm cell I already told that for movement, then nerve cell for the transmitting nerve impulse, muscle cells which are cylindrical and they have got on alternate light and dark bands. So they help in the contraction and relaxation and the coordinated movement of our body along with the skeletal system. And also the bacteria which are of different shapes like the rods, then the spherical, the spiral. So therefore the bacteria shapes actually have the major variety into the four types. One is the round shape, which is known as coccus. The rod shaped is known as the bacillus. Then we have got the spiral and the spherical shape is that a round shape only, which is known as the coccus. The rod shape is known as the bacillus. Spiral shape is known as the spiralium, and the another one is comma shape. Just like the comma, it is known as the vibrio. These are the four major categories of bacteria based on their shape. So these are the pictorial representation of various types of cell shapes based on their functions. Now coming to the size, we have already watched in the video that the largest egg the largest cell in the living world is that of the ostrich egg and the size is this, right? Then we have got the smallest cell which is of a bacteria called the mycoplasma and this is also known as the PPLO or pleuropneumonia like organisms, right? This is the smallest type of cell in the living world. The longest is the neuron. And if I talk about the human body, in the human body, the longest cell again is that of the neuron. The largest cell is of the ovum which is produced by the ovaries in the female reproductive system, and the smallest is that of the sperm which is produced in the male reproductive system. Now we'll watch a video regarding the prokaryotic and eukaryotic cells. So first we'll watch a video and then we'll discuss what they are and what is the difference between the two types of cells.
Cells can be differentiated in many ways. You're right. There is no boundary that encloses the nuclear material. This undefined nuclear region contains only nucleic acids. What are nucleic acids? Have you heard this term before? Nucleic acids are the genetic material of the cell and are made up of DNA and RNA. Both these help transfer the genetic traits to new cells. This nuclear region with the nucleic acids is called the nucleoid. What about the plant cell? Is its nuclear region undefined as well? No, the plant cell has a clear, distinct nucleus. This nucleus has a nuclear membrane, and all the nuclear material is enclosed within the nuclear membrane. The cells which do not have a well-defined nucleus are called as prokaryotic cells. What does prokaryotic mean? Pro means primitive, and karyon means nucleus. Therefore, a prokaryotic cell has a primitive nucleus which is not well defined. On the other hand, the nucleus of the plant cell does have a nuclear membrane. This is a well defined cell. The nuclear material is separated from the cytoplasm by the nuclear membrane. What are these kind of cells called? They are known as eukaryotic cells. Eu means true and karyon, as before said, means nucleus. All organisms, other than bacteria and blue-green algae, are called eukaryotes. What other differences did you notice in these cells? The prokaryotic cell does not have these cytoplasmic bodies. Can you see the membrane-bound organelles in the plant cell? Does the bacterial cell have them? No. The prokaryotic cell lacks membrane-bound organelles. Then who carries out the cellular functions for the prokaryotic cells? As we can see, the cell does have some poorly organized parts. These carry out the basic functions of the cell. However, these parts aren't as well defined as the eukaryotic cell organelles. What are the major differences between a prokaryotic and a eukaryotic cell? The prokaryotic cells are smaller in size, unlike the eukaryotic cell, which is comparatively larger. Do you know a prokaryotic cell size ranges from 1 to 5 micrometers, whereas the size of a eukaryotic cell ranges from 10 to 100 micrometers. The prokaryotic cell lacks a well-defined nucleus and membrane-bound organelles, unlike the eukaryotic cell, which has both of these. Generally, a prokaryotic cell contains only a single chromosome, which is a thread-like structure that contains the genetic material in the form of DNA. The eukaryotic cell has many chromosomes enclosed in the nucleus. With this, we clearly know the difference between a prokaryotic and a eukaryotic cell. We will study the organelles in details in our coming videos.
So let's summarize again, though it has been clearly stated in the video. So for our revision purpose, let's summarize. So prokaryotic means you have already now understood from the video, I guess. Pro means primitive and karyon means the nucleus. So the primitive type of nucleus which actually does not contain any clearly defined structure such as the nuclear membrane, nucleolus, then the chromosomes arranged in a particular order, then the nucleoplasm. So all these structures means a well defined nucleus. So those are the organisms which are called prokaryotic cells or prokaryotes, and the examples are the bacteria and the blue green algae. And why these are called prokaryotes? Because when life first came on earth, these were the first type of cells that formed when from the inorganic substances. So from the postulates of cell theory, we know that cells always arise from the pre-existing cells. But the question here is, when did the first cell and how did it come to life on earth? So therefore, here also you will study in higher classes, the experiment was conducted and there it was confirmed that the first cell, which is the organic cell in the form of prokaryotic cell, first arrived in the oceans, that means in the sea water, and also it came to life from the inorganic substances. And actually the environment that existed on earth during that time, so that gave rise to the birth of the cells or the primitive types of cells having the primitive type of nucleus. So you can now imagine, so according to the age of first life which on this earth, so that is the age of bacteria and blue green algae. So from that time they are existing along with the first there were only them which existed, and later when eukaryotic cells developed, so now they are coexisting together on earth. So therefore this is the primitive type of nucleus or primitive type of organisms having such kind of simple nucleus. On the other hand, U means true and karyon again means nucleus. So the true nucleus consists of many parts such as the nucleolus, then nuclear membrane, nucleoplasm, and many chromosomes. In case of prokaryotes, we have only a single chromosome which is spread in the cytoplasm of the cell, but in case of the eukaryotes, we have a definite place, that is the nucleus, within which chromosomes are properly arranged. So these are the differences, the main primary difference. But apart from them, you have already watched other differences, such as, since these are very primitive type of cells, so they do not need to conduct the complex processes. So that is why they do not have the cell organelles to actually follow the mechanism of division of labor. So they have the no cell membrane bound, or they have no membrane bound organelles such as the mitochondria, then the plastids, then the lysosomes. They do not have any of them. And on the contrary, we have all of them in the eukaryotic cells, that we can see a lot of them in our surroundings. Also, in case of the prokaryotic cells, since they were very minuscule in their structure, so therefore their organization as well as their size is very small as compared to the eukaryotic cells. Now eukaryotic cells actually have larger size, that also you have watched in the video, and their organization is also far more complex as compared to the organization of the cell organelles, and also organization of the cells also to form the hierarchical order in order to give rise to multicellular organisms. So therefore in both ways eukaryotic cells are way more complex as compared to the prokaryotic cells. So again, these are the postulates of cell theory which we have already discussed. These are the scientists who contributed to the postulates and we have already discussed them. Now coming to the plant cell and animal cell. So what are the basic comparative or comparisons between the plant cell and animal cells? So we already know plant cells, the distinctive feature of plant cell is the cell wall. So whenever we see any cell that contains a thick cell wall or outer covering, or it has got a rigid or structure, definite structure, so we can then discern from the observation that this is a plant cell. But on the other hand, if we see that we can see only a thin membrane and no such particular structure, only a circular type of or oval type of structure, then we can decide that it is the animal cell. So therefore, apart from this also, there are also differences in other parts or other cell organelles. So let's watch the video regarding that.
In our previous video, we looked at a plant cell. You now know how to mount a microscope slide, don't you? We will now look at an animal cell. You will once again require a few things: a microscope, a slide, methylene blue, and a toothpick. Add a drop of methylene blue. Now gently scrape the inner side of your cheek with a toothpick. Dab the tip of the toothpick onto the slide. Let us observe this under the microscope. What are we observing here? That's right. We are looking at cheek cells of humans. These look so much like the plant cells. Hold on. We will watch it a little more closely. Do you remember there are two distinct boundaries around the cell? Yes, the cell wall and the plasma membrane. Notice that we do not have the cell wall around the plasma membrane in the animal cell. Okay, how about we do one thing? Let's understand the animal cell by comparing it with the plant cell. There are many other differences. Can you spot them? In the plant cell, there is a large empty structure and it's called the vacuole. In the animal cell, there are several smaller ones scattered all over the cytoplasm. Let me repeat. The plant cell has one single vacuole, while the animal cell has several smaller ones. Also, in the plant cell, there are several colored bodies. Do you know what they are called? They are known as plastids. These are absent in animal cells. Look at this plastid in the plant cell. It's called the chloroplast and it contains chlorophyll. This pigment gives the green color to the leaves. I'm sure you know the other purpose for which chlorophyll is used. Yes, plants require chlorophyll for the process of photosynthesis. Let us review the differences between the plant and the animal cell. The plant cell has a cell wall, whereas the animal cell doesn't have it. The plant cell has one big single vacuole, whereas the animal cell has several smaller vacuoles. Lastly, the plant cell has plastids, while the animal cell does not. Apart from the nucleus, vacuoles, and plastids, there are several other organelles in the cell. You will study them in our other videos. The entire content of a living cell is called as protoplasm. What is protoplasm then? Protoplasm is the living substance of the cell. It includes the cytoplasm and the nucleus. Can cells be differentiated in other ways? We'll see that in our next video.
So therefore, the basic differences in the plant cell and the animal cell has been described in the video. So apart from them, I will now discuss some other differences. For example, the lysosome. which is also known as the suicidal bag, and about which we will study later on in this chapter. So lysosomes are actually, they are very prominent in case of animal cells, right? Because they do not contain the vacuoles, but they are either absent altogether, or if present, very rare, but not so much that in plant cell, because the plant cell already has got a large vacuole which occupies almost 30% of the mature plant cell, and it carries out the functions of the lysosomes also. So therefore there is no further need of additional lysosomes here. But in case of animal cells, since it lacks the vacuole, so it needs the presence of lysosomes. So here we have the presence of lysosomes, and here we can say lysosomes are absent. Now another thing is that the nucleus, since the vacuole occupies the major portion in a plant cell, so the nucleus in case of plant cell is pushed towards the periphery, of the cell. But in case of animal cell, the nucleus is present almost centrally, central nucleus. Right. So, and where we can say periphery nucleus. So, and apart from them, the other organelles which have been discussed about the plastids. Yes, plastids, when we will study, we will discuss about the categories of plastids and their functions also, and then we will understand why they are present only in plant cell and not in animal cell. So total we have got the 5 points of differences. One is the cell wall, then second thing is the presence of and absence of vacuole, then third thing is the presence and absence of lysosomes, then fourth thing we have got about the peripheral and the central nucleus, and also another point which we have got from the video also, that the plastids which are present in the plant cell but absent in the animal cell. So in plants we generally have the root cells which absorb the water and the minerals from the soil, and if the root cells die while plucking the sapling, if the root tip or the roots are damaged in some or the other way, then the plant wilts and dies, because then the root cells that are damaged and cannot further transport the water and the minerals, without which the plant cannot survive or carry out the process of photosynthesis. Then the leaf cells also, just a moment. And sorry for the interruption. So now again for leaf cells, so they actually manufacture food and contain the chlorophyll, which help them to absorb the solar energy or the light energy of the sun, and then it helps to convert that solar energy into the chemical energy of the food, that is the glucose, which is prepared as a result of photosynthesis. And now in case of animals, we have got different types of cells such as the muscle cells, which enable the movement of various parts of our body by contraction and relaxation. Either the muscles move according to our own will or according to the involuntary movement which is required, which cannot be controlled by us. Then we have got the nerve cells which conduct and transmit the neural messages from one end of the body to another. And then we have the gland cells which secrete the enzymes and the hormones for the proper digestion and also for the proper messaging or the signaling system of our body. And we also have got the skin cells which forms the outermost covering of our body and protects us from the wear and tear, damage, as well as invasion of various types of pathogens present in our environment. That is why, and it also helps in regulating the body temperature by actually forming the sweat, which evaporates and leads to the cooling down of the body temperature. So these are all the functions of various types of cells present in our body. Now we'll discuss and move on to the next portions, that are the structure and components of the cell and also the cell organelles in detail part by part in our next lecture. So thank you so much for staying with us. I hope you have got an idea about the basics of the cell, and again if you have any doubt, do let us know in the comments below. Thank you so much. Take care. Bye bye.